Hello and welcome to the second video. We're going to be looking at cell division, focusing mainly on mitosis, a little bit stem cells in here as well. The specification reference is 4.1.2. So this video is going to look at chromosomes, the cell cycle with a little bit of mitosis in there, or quite a bit of mitosis in there, and stem cells, including the ethical dilemmas, and we'll finish off looking at a long answer exam question and how I've had a go at answering it. So we'll start off looking at chromosomes. So on the right of the screen we've got four words: DNA, gene, chromosome and genome. You need to know what they all are. I suggest you have a go at writing down definitions or explanations of them. I will reveal the answers later in the video. This is also just to keep you watching. So we've got chromosomes. So DNA is the chemical that makes up our genetic material. It stands for deoxyribonucleic acid. We've got a nucleotide base, we've got phosphate, and we've got sugar all in there together. It makes that lovely double helix structure, that sort of swirly structure going round. It's got two sides joined together in the middle. And we sort them out into long strands called chromosomes. We've got very long strands of DNA called chromosomes. Humans have 23 pairs of chromosomes, one on each side of that double helix. We get one from our mother and one from our father, or half from our mother rather and half from our father. So 23 single chromosomes from each, which makes 23 pairs. Now our chromosomes contain genes, and genes code for different proteins. And it says there they're sorted into pairs, and humans have 23 pairs of chromosomes. Now this bit here, we've talked about the genes coding for proteins. This is going on to the essential dogma of biology, which is that DNA codes for RNA and RNA codes for proteins. That is essentially biology. Everything else is just developed from there. So at the bottom I've got a picture of some chromosomes. We've got, for human chromosomes, we've got chromosome 1 all the way up to 23. Now you'll notice that they are all numbered, apart from number 23, which is just X and Y. That is the sex chromosomes. So in this case the person is male because they have that Y chromosome. You'll notice the Y chromosome is a lot smaller than the X chromosome, probably because men are missing something compared to women. Those of you that are in my classes should be aware, hopefully, that I do think women are pretty much better than men in every single way possible. So moving on to the cell cycle. So the cell cycle is the life cycle of the cell. It's a bit like the circle of life. We've got the carbon cycle we'll look at later on. Now they can show you various different diagrams, but all they'll want you to do is identify which part of the cycle the cell is in, or how long it's lasting for. If you're working out how long it's lasting for, you'll need to find the proportion of the circle that it lasts for and work that out as a percentage of the total time. This is using your maths skills. I'm not going to step on any math teacher's toes and explain how to do this at this point. So the cell cycle can happen very quickly or very slowly. It happens very, very quickly during the first few months of life. So that fertilization, when that egg meets the sperm, to birth, actually all through childhood as well. The cell cycle is happening very, very quickly. We are growing at a very, very fast rate. As we reach adulthood, it begins to slow down. But even in adulthood, some cells will grow faster than others and will go through that cell cycle faster than others. So for example, red blood cells, their cell cycle, or their cell life, lasts for between 80 to 120 days, whereas other cells will go very, very quickly, very, very slowly rather. So stage 1, we are in interphase. That's that big stage. Now this is the normal cell activities where the cell just gets on doing what it wants to do, whatever its specialization is. Towards the end of it, the cell starts getting bigger. It starts growing in size. It will also synthesize more cell organelles, so things like the mitochondria, those ribosomes, the Golgi apparatus. And it will also do this bit here of duplicating the chromosomes. So we're duplicating the chromosomes so that we can, when we split and make two daughter cells, they will have the correct number of chromosomes. So duplicating, we're making exact copies. They are identical copies. You can see there on the image we've got a cell that is getting larger. Its nucleus is getting a lot bigger. It's growing in size. We can tell that is in those last few stages of interphase. So next up we've got stage 2, which is mitosis. This is where the magic happens. It's that cell division. So the nucleus will divide into 2 separate nuclei. They pull apart. The chromosomes have all lined up in the middle and are being pulled apart. We've got 2 separate nuclei forming. We'll have a look at a picture of one now. There we go. We can see those nuclei on that middle cell being pulled apart. You can see the chromosomes being pulled apart. Also, it's worth noting that when we're looking at this happening, the chromosomes become a lot larger and a lot thicker, meaning that we can see them under a light microscope. If we look at the cell to the right of that one in the middle, you can see we've got those 2 nuclei formed around it. They're at separate ends of the cell. We've got those things forming. Next stage we've got is the cell dividing in 2. This is called cytokinesis. So our cell has divided in 2. We've got those 2 nuclei, but at the moment it is still just one cell. There's only one membrane around them. There's nothing separating them. But they contain enough organelles for 2 cells. So we get a new cell membrane forming between the 2 cells. We've got a picture there, you can see in the middle, we've got that cell membrane, or cell wall and membrane in this case. This is an onion root tip cell. We can see it there forming between the pair of them. So there we go, we've got the cell cycle. We've got stage 1 interphase, stage 2 mitosis, and stage 3, which is cytokinesis. You need to be able to identify which stage they are in. So the cell cycle is very, very important. It allows us to grow. We can get bigger, fatter, stronger, grow as organisms from a single cell into the multicellular thing that we are today. Also allows us to repair tissues. If you cut yourself or get a graze on your leg, it heals by mitosis. We've got those skin cells are healing by mitosis. That's what our growth is, and that happens fairly quickly, as you can see as well. We also are able to develop as our multicellular organisms. We can get bigger. Exam tips. So things you may be asked in an exam. You may need to identify what cell is undergoing mitosis. So they may show you a number of different cells and ask you to say which one is undergoing mitosis, or indeed which one is in interphase, or in which one is in cytokinesis. So looking at what is happening to the nucleus. If the nucleus is just there by itself doing absolutely nothing, it is probably in interphase. If we've got 2 nuclei forming, so being pulled apart, we're going to be in that mitosis phase. And if we've got 2 cells being formed, we're in cytokinesis. So we need to know as well that mitosis needs a lot of energy. It takes a lot of energy to make all these new organelles and to pull chromosomes apart, or duplicate those chromosomes, pull them apart and make a whole new cell. We get that energy from mitochondria. We'll also need to be able to compare mitosis and meiosis. Now this is more likely to come onto paper 2 content, as this is where we study meiosis, which is that sexual reproduction. So you may remember at the start we looked at DNA, gene, chromosome and genome. Here are the definitions for them. So DNA is the chemical that makes up our genetic code. The gene is a section of DNA that codes for a protein, so we're moving up in size. DNA, the smallest one. Gene, a section of DNA, a number of DNA molecules. Chromosome, that is a number of genes. We've got a number of genes together, make a chromosome. All of the chromosomes together, 23 pairs in humans, make up our nucleus. Obviously all nuclei in humans are identical. We get that genome at the end, which is the entire genetic material of an organism. Think about the Human Genome Project we had a few years ago, that we do teach. It was finished quite a few years ago. I remember it happening when I was at school, finishing. Quite exciting. They've mapped the entire human genetic code, leading to lots of possible exciting things. So, last thing we're going to look at is stem cells. So, stem cells are undifferentiated cells. that can become any specialised cell within that organism. Now, they will differ between animals and plants. Animals are a lot more basic than plants when it comes to this. So, animal cells, we've got two different types. We've got embryonic stem cells and adult stem cells. A bit of confusion happens here sometimes where people think that an adult stem cell has to come from an adult, someone over the age of 18. It doesn't. It is just from something that is not an embryo. Those of you doing child development will obviously be aware that an embryo is that those fertilized eggs in those first few days when nothing yet has formed. It's just a ball of cells. Now, those cells in those embryonic stage are quite special. They can differentiate to become any type of specialized cell within a person. There's a lot of controversial issues with this because we can't ask for consent to take them from the embryo. We can obviously ask the parents, but we can't ask the embryo itself. There is a very, very large risk that we may cause death or permanent harm or disability to the embryo as it develops into a child, or a baby and a child. But it can become any particular cell, particularly nerve cells, which find it very, very hard to regrow. We don't have nerve stem cells. Therefore, if people have nerve damage, they often cannot walk in. So if they have a broken back and we break the spine, they are unable to walk again. Potentially embryonic stem cells give them that chance to walk again. The other type of stem cells we have for humans or for animals are the adult stem cells. These are often taken from the bone marrow or the liver per se. We've got different types of them, and they can only make a certain kind. So if we took a liver stem cell, it could make any liver cell we wanted, but it couldn't make a blood cell or a skin cell. If we took bone marrow stem cells, we can make any type of blood cell, so red blood cells, white blood cells, but we couldn't make a liver cell from it. Now obviously with these, they are a lot easier to take. You can get consent, very, very unlikely to cause harm, but it does cause an awful lot of pain. If someone donates their stem cells, they get a very large needle put into their hip bone, that hip joint, and take out an awful lot of it. We've now got the plants, which have got the meristem. So this is where we get our stem cells from plants. It's in that meristem. They are undifferentiated plant cells, and it can become any type of the plant. So it can become a root cell, a leaf cell, part of the xylem, part of the phloem. It can become any part, as opposed to adult stem cells, which are limited. Now, this is very, very useful for plants. It can allow us to save rare plants. We can take a plant that is very, very rare, is dying out, and we can take some stem cells and grow whole new plants from it. We can do lots of investigations by using genetically identical plants. It's quite useful for science and researchers. Can also use it in horticulture and agriculture to produce those large numbers of crops that have certain characteristics such as disease resistance. So the last bit we're going to do is have a go at this 6-mark question on stem cells. So there is a paragraph there for you to read, and then a question at the bottom in that yellow box, which is asking you to evaluate the use of stem cells from embryos or from adult bone marrow for treating human diseases. You should give a conclusion to your evaluation. And what I suggest you do is read through this paragraph, pause the video for about 5 to 6 minutes, and have a go at answering the question. I will then show you my answer, which I have included more than is needed in it, just to give you an idea of the things that we're wanting. So I'll now be moving on to my answer to show you what I've got on there. So this is my answer. I will reveal the whole of it. You'll see that going through it, I have given pros and cons for each one. I've compared them. I've discussed the same point for each one. So the starting point, I have looked at how the cells can differentiate. Embryonic stem cells can become any kind of specialized cell, but adult cells can only become one type and can only treat a certain disease. I've looked at the pros and cons for each point, and at the end I have given a conclusion basing it on my thoughts and beliefs. Now I've tried to stay away from religious points of view, so we cannot say, for example, that I think it is religious. I think it is ethically wrong, and leave it at that. If you give a point for your answer, so I think it is ethically wrong, or morally wrong for religious reasons, because the embryo is unable to give consent, because we're causing harm or risk of death to the embryo, then that is fine. But we cannot just say we object for religious reasons or moral reasons without backing it up. We need to have a rationale for our choice. Okay, thank you very much for watching this video. I hope it is informative. Next week I will look at doing cell transport, so that's osmosis, diffusion and active transport. Please subscribe, click the link that is somewhere around the screen. Please subscribe, that way you get updates. If I get to 100 subscribers, it means it's a lot easier to find the page as well. Thank you very much.